Hi, I'm Tonia Korves from MITRE. I'm presenting a tool to help with the large volume of COVID-19 research. It's called the COVID-19 Therapeutic Information Browser, or COVID-TIB for short. We built COVID-TIB to make it easy to find and track information about potential therapeutics and vaccines. COVID-TIB is a resource for finding publications and clinical trials about viruses and therapeutics. It includes candidate drugs for SARS-CoV-2 and 12 other viruses. It also includes information about viral vaccine research. And to facilitate tracking the status of research, in COVID-TIB results are sorted by research stage, such as clinical study, case study, or in silico. Now, to capture this information at scale, we applied NLP. There are challenges in building an NLP pipeline during a pandemic. First, it must be built very quickly because it's needed right away. We had no prior curated data available for training an algorithm. And the system needed to be adaptable for a rapidly evolving research landscape. Here's our NLP approach. First, we query for viruses and retrieve documents. Our sources are PubMed, COVID-19 for preprints, and clinicaltrials.gov. Then we use a two-step rule-based NLP approach. First, we identify entities and keywords within sentences. And for this, we modified Reach software from the University of Arizona. We use custom dictionaries for viruses and drugs. There are 12,000 different drugs in our dictionary, most with drug bank IDs. We also use custom keyword lists for research stages. Then we assemble information across sentences and classify documents. For this, we use R scripts with rules and threshold of evidence for classifying documents as about a particular virus and drug and in a given research stage. Now we iteratively and incrementally improve the NLP results over time. And for precision, we did this through curation. In the early stages, we used a curation platform that we developed to identify errors and then make improvements in rules. For recall, we made improvements by comparing to clinicaltrials.gov and lit COVID results. To date, we've processed more than 270,000 documents. We've identified 26,000 paper abstracts and clinical trial summaries about SARS-CoV-2 and named therapeutics. And we've identified documents about nearly 3,000 different therapeutics. We've also incorporated full text search and information retrieval capabilities using two tools from the University of Wisconsin-Madison. These are XDD, which is a searchable digital library with full text publication content provided by open access and partner publishers, and Cosmos, which is a machine learning-based platform designed to serve as a technical assistant for extracting information from documents. Now I'll give you a demo of COVID-TIB. This is the COVID-TIB homepage with a description. On the left is a navigation menu. There's a therapeutics page and a vaccine research page. Let's click on therapeutics. Here we see a table with drugs going down the side, viruses across the top, and research stages beneath that. And the numbers in this table indicate the number of documents about a particular virus, drug, in a particular research stage. So we have 372 documents about remdesivir and SARS-CoV-2 that are clinical studies. We can expand the list of drugs or scroll over to see additional drugs, and we can also scroll over to see additional viruses. Let's say we want to see the drugs that have been published about recently in silico studies. We can go over here to the filters. We can go ahead and select past 2 weeks, and then we can go ahead and sort by in silico and see those drugs. Now let's say we want to look for a particular drug. We can go ahead and type the name in here. Let's look at famotidine, and let's select clinical studies. Then if we scroll down, we get a table that shows the documents here with link outs. And we can also download this list of documents as a CSV file here. We can also go ahead and link out to Cosmos for figures and tables about SARS-CoV-2 and famotidine from papers. Here on the Cosmos site, we see related terms and we see figures and tables from papers. Say we want to see drugs for long COVID. We can go ahead and type the long COVID in as well as some synonyms. Then go ahead and search for these, and what it's doing is searching for these terms in the full text of a subset of the papers. And here we see the drugs with papers with these terms in it. We can go ahead and click on the one for glucocorticoids. And then if we go ahead and scroll down to the bottom in our list, we now have the option to search snippets. So we can go ahead and click on this, and what it does is it shows us the bits of sentences from the paper with our search terms in it. There are additional files for download on the menu, including data, information about our methods, and an analysis of COVID drug research trends using this data. In the Biocreative Evaluation, there were 12 users. They commented that it was easy to use, fast, and well organized. They suggested improvements in adding reset buttons, in improving ability to find drug names, and an increasing amount of data for download. And their comments also highlighted that comprehensiveness is particularly important in a tool like this. And this is an outstanding challenge for entities like drugs, which have rapidly evolving namespaces. COVID-TIB has multiple potential uses. The Biocreative users suggested that it could be useful for knowing the state of research, for systematic reviews, and for clinical guideline development. To date, researchers have used COVID-TIB to gather information for drug research studies, to identify papers for a curation effort, and to identify test cases for the MI models that you saw earlier. COVID-TIB also has potential applications beyond COVID-19. It could be adapted to track research on therapeutics for other viruses and diseases. And more broadly, the approach could be applied to research in other domains that also have a large and growing literature. With that, I'd like to acknowledge my colleagues who contributed to this work, who are listed here, and thank our funding sources. For additional information, please see our website. Thanks.